Hello students, I am Dr. Deepika Paliwal. Welcome you all in this virtual class on the behalf of School of Medical and Allied Sciences, Belgorod University. Today we will discuss alkylating agent, a category of the anti-cancer agent. Prerequisites. Before we start our lecture, the student must be aware about the term structure of nucleotide base and structure of DNA. You already studied this topic in your previous class. Objective of today's lectures, the student will learn about the chemical classification of alkylating agent. We will study different categories of the alkylating agent, differentiate the receptor binding pattern and the structure feature needed between agonist and the antagonist, different approach in getting anti-cancer agent. Introduction of the alkylating agent, oldest and most useful of anti-neoplastic drug, directly act on the cells that are they also known as cytotoxic drugs. Clinically used evolved from the observation of bone marrow suppression and the lymph node shrinkage in the soldier exposed to sulfur mustard gas warfare during World War I. These are, these are first used in the First World War. Effectiveness as the anti-cancer agent was confirmed by clinical trials in the middle 1940s. The alkylating agents are a class of drugs that are capable of forming covalent bond with important biomolecules and prevent the replication and transcription process of mechanism of action of alkylating agent. Alkylating agent produce carbonium ion that are highly reactive. When alkylation is done, these transfer the alkyl group or substitute alkyl group to the cellular macromolecules by forming the covalent bond. These macromolecules are basically the nucleotide base of DNA. They are basically act on 7th position of the guanine. Then after the alkylation, they make cross-linking and abnormal base pairing or scission of the DNA strand. This will lead to the inhibition of DNA synthesis. This is the inter-strand cross-linking by alkylating agent, and this is the inter-strand cross-linking. Classification of alkylating agent. Alkylating agents are classified into basically 4 categories. Nitrogen mustard compounds. Under this category, cyclophosphamide is drug. Then ethylene amine. Thiotepa belongs to ethylene amine category. Alkyl sulfonate. The drug comes under this category is busulfan. Nitrosourea. The drug comes under this category are carmustine and lomustine. So these are the 4 important classes of alkylating agent. Nitrogen mustards compound. The first category of alkylating agent. These compounds developed from the mustard war gases that is used in World War I, which are highly reactive vesicants. These agents used for cancer. These agents are not cell cycle specific. They do not particularly act on a particular phase, but still more active in dividing tissues. They are radiomimetic, means their action on DNA resemble to the radiation. The drug comes under these categories are meclorethamine, chlorambucil, melphone and cyclophosphamide. These are the structure of these drugs. Mechanism of action of nitrogen mustard compound. Take an example of the drug that is a chlormethamine, which on reaction give a azridine ion upon the removal of chloride ion. This azridine ion attack on the 7th position of the guanine and they make a covalent bond with DNA. After that, the other chloride ion is removed and make a azridine ring and they also act on the guanine and make a covalent bond with the DNA. This they alkylating agent then cause intrastrand and interstrand cross-linking. This will prevent the replication of the DNA. Monoalkylation of the guanine is also possible if there is one electron withdrawing group is there. So this cross-linking will result in the inhibition of DNA synthesis. The second category of alkylating agent is ethylene amine and the drug belongs to this class is thiotepa, that chemically known as triethylene thiophosphoramide. It is an organophosphorate compound with the formula SpNC2H4 whole thrice. This molecule features tetrahedral phosphorus and it is structurally akin to the phosphate ion. It is derived from the azridine and triphosphoryl chloride. Look at the mechanism of action. This is the thiotepa. On hydrolysis, give one azridine group free and this azridine group make a covalent bond with the nucleotide base of the DNA. This is the another pathway where the thiotepa azridine ring is opening and that make a covalent bond with the 7th position of the guanine which is a DNA monoadduct. Same like the other azridine are also open and they make a covalent bond with the DNA. This cross-linking will result in the inhibition of the replication process of DNA and inhibit the DNA synthesis. Thiotepa is used in the treatment of adenocarcinoma of the breast, adenocarcinoma of the ovary, superficial papillary carcinoma of the urinary bladder. The third category of alkylating agent are alkyl sulfonate and the drug belongs to this class is busulfan, chemically known as butane 1,4-diyl dimethane sulfonate. Busulfan utilize two sulfonate functionalities. These are the two functionals group as liming group separated by a 4-carbon chain that react with DNA to primarily form the intrastrand cross-linking. Alkyl sulfonamide used as a chronic granulocytic leukemia and in the bone marrow transplant. Look at the mechanism of action. This is the busulfan which on decomposition gives the carbocation. This carbocation alkylate the 7th position of the guanidine, that is a monoadduct of DNA. The monoalkylation was done and then this alkylation was done and they make a intrastrand cross-linking with the DNA. The last class of alkylating agent is nitrosourea. They are the derivative of nitro and the urea. These are unstable in the aqueous solution. They produce isocyanate and a diazohydroxy upon decomposition. Nitrosourea derivative decompose to give isocyanate, that is the carbomylating agent, and a nitro diazo compounds. These diazo compounds on decomposition gives the alkylating agent a carbocation which on DNA alkylation to give the cross-linking between DNA. These type of alkylating agent cause interstrand cross-linking between guanine or the guanine cytosine. Carbohylating agent react with lysine residue on the protein that may inactivate the DNA repair enzyme. Now proceed to the practice question. These are very important from the examination point of view. Look at them. Discuss the mechanism of action of mechlorothamine. What are the different chemical classes of alkylating agent? Discuss or what is the mechanism of action of nitrosourea? So prepare for this question. Dear students, if you want more information regarding this topic, you can refer to these books. We will meet again in the next lecture. Till then, take care. Namaste.